Okay, so let's dive into these longevity papers.
Mhm.
This week, we're looking at a whole bunch of research papers published specifically, for biotech researchers.
Yeah.
Our focus this week is on the papers that were published March 3rd through the 6th,
Yeah.
really honing in on the papers that are really surprising. They're
Yeah.
coming from credible institutions.
Mm.
The type of research that, is going to make us maybe rethink,
Yes.
how we understand aging.
Exactly. We're talking about model change here.
Yes.
Papers that are really going to move the needle in terms of how we think about aging and how we do research in this space.
Absolutely. So we're not going to just list every single paper. We're going to deep dive into a few and
Yeah.
really unpack
Mm-hmm.
what's so remarkable about these papers
Absolutely.
and, what implications they may have for,
Yeah.
future research and development.
Yeah. And the application of these findings
Right.
to longevity biotech.
Yeah. So are you ready to kind of explore some of these papers?
Let's do it.
Right.
I'm really excited about a paper that came out just yesterday, March 6th, in tumor suppressor protein P53 from Sanford Burnham Prebys Medical Discovery Institute. The title is P53 enhances DNA repair and suppresses cytoplasmic chromatin fragments and inflammation in senescent cells.
Okay. Break that down for me. What's so special about this P53 paper?
Well, so as you know, cellular senescence is a hallmark of aging.
Right.
It's basically where cells stop dividing
Yeah.
and start pumping out inflammatory signals.
Right. They're kind of like they've retired.
Right.
And they're just causing trouble.
Exactly. And
Yeah.
so the thinking has been that senescence is a one-way street.
Right.
Once a cell becomes senescent, there's no turning back.
Right.
But this research really challenges that assumption.
Okay.
They found that P53, which is known to be a tumor suppressor and involved in DNA repair, can actually reverse some of the key features of senescence.
Wait, reverse senescence. So that's huge. How does that even work?
They found that by activating P53 in aged mice, they could actually enhance DNA repair in senescent cells.
Okay.
And that led to a decrease in the formation of these inflammatory fragments called cytoplasmic chromatin fragments.
So it's kind of like cleaning up the debris in the cell that's contributing to the inflammation and the aging.
Yes. Yeah.
So are they kind of like giving those retired cells
Yes.
a second chance to be productive?
Yes. In a way.
Okay.
They saw a decrease in the accumulation of certain immune cells in the liver and even a reversal of some age-related changes in gene expression.
Wow.
Now, this is just in mice, but if these findings translate to humans, it could have really big implications for developing therapies that target P53 to actually reverse some of the damaging effects of aging.
This could be huge for treating age-related diseases.
Mm-hmm.
But I got to ask, what about the risk of, activating P53? Isn't it also involved in cancer?
That's a great question, and that's something that researchers are going to have to look into.
Okay.
There's always a balance when it comes to manipulating biological pathways. But the potential benefit of being able to reverse senescence is so great that this research definitely needs more exploration.
Right.
It could lead to entirely new therapeutic approaches for not just treating age-related diseases, but potentially even rejuvenating tissues and organs.
This is one to watch, for sure. As a biotech researcher, you're probably already thinking about the applications of this.
Absolutely.
Yeah.
And speaking of rejuvenation, there was another paper this week about stem cells and their potential to combat aging.
Yeah.
This paper came out on March 5th in Cell Biology by a team at Hanyang University, and it's titled Human Hypothalamic Neural Stem Cells Transplantation Ameliorates Age-Associated Dysfunction Through SNC and MPT Axis Activation in Aged Mice.
Okay, lots to unpack there. First of all, just remind me what's so important about stem cells when we're talking about aging.
Sure, stem cells are the body's repair crew.
Okay.
They have this unique ability to differentiate into various cell types, replenishing tissues and organs as they wear down over time. But as we age, the number and function of stem cells decline.
All right, so it's like our repair system starts to falter.
Exactly.
Okay.
And that contributes to a lot of the deterioration we see with aging.
Got it. So what did they do with stem cells in this study?
Well, they focused on a specific type of stem cell called hyperthalamic neural stem cells.
Okay.
The hypothalamus, as you know, is this tiny region in the brain that plays a really big role in regulating a wide range of bodily functions, like hormone production, sleep cycles, appetite, all sorts of things.
So it's like the control center.
It is.
And as we age, that starts to malfunction.
Precisely. And that can have cascading negative effects throughout the body.
Right.
So these researchers wondered, what if we could replenish these hypothalamic stem cells and restore function to that crucial control center?
Bold idea. Did it work?
The results were actually pretty amazing.
Okay.
They took human hypothalamic neural stem cells
Okay.
and implanted them into the brains of aged mice. And what they found is that those mice actually showed improvements in a bunch of areas, including metabolic fitness, physical ability, and even cognitive function.
Wow, a triple threat.
Yeah.
It sounds like those stem cells really rejuvenated that hypothalamus.
It's quite impressive.
Yeah.
And they didn't stop there. They wanted to understand the mechanism behind those improvements.
Okay.
They discovered that the transplanted stem cells triggered this cascade of events that boosted the production of a molecule called ENA-MPT in the body.
Okay.
And ENA-MPT has been shown to have anti-aging properties.
Right.
And it's involved in energy metabolism and cellular repair.
So it's not just replacing the cells, it's also stimulating the production of these beneficial molecules
That's right.
that could have a system-wide effect.
Exactly. So this research opens up all these new possibilities for targeting the hypothalamus as a way to combat aging.
Wow.
You know, imagine being able to develop therapies that either replenish those hyperthalamic stem cells or boost ENA-MPT production to really revitalize this crucial control center in the body.
It's like giving the body a software update,
Yeah.
improving the operating system itself.
Yeah.
I'm sensing a theme here. First, we have a paper showing that we might be able to reverse senescence.
Yeah.
And now we have research that says we can rejuvenate the control center of the body.
Mm-hmm.
It seems like we're moving beyond just slowing down aging and starting to talk about actually reversing some of its effects.
I agree. There's a lot of optimism in the field right now. And then this next paper adds to that feeling. This paper focuses on the brain, specifically how cells respond to stress and how that response might be linked to lifespan. This one was published on March 4th in Cell Biology from the University of Miami Miller School of Medicine.
Okay.
And the title is Control of Stress-Activated CDC42 Dynamics by the MAP kinase STI1-NDR kinase ORB6 regulatory axis.
Okay, before we dive into that one, can you just give me like a quick refresher on why cellular stress is relevant to aging?
Absolutely.
Yeah.
So our cells are constantly exposed to stress from, things in the environment, toxins, to internal metabolic byproducts.
Right.
And over time, all of the stress takes a toll.
Right.
It leads to damage.
It's like wear and tear.
Yes, wear and tear at the cellular level.
Right.
And that contributes to aging and disease.
Got it. So how does this paper fit into that?
Okay, so they were looking at this protein called CDC42, which plays a role in cell growth and division and how cells respond to stress.
Okay.
And what they found was that when cells are under stress, like when nutrients are scarce, a specific signaling pathway involving these two enzymes called STI1 and ORB6 influences the activity of CDC42.
Got it.
And that helps cells adapt and survive.
So STI1 and ORB6 are like messengers.
Exactly.
Helping coordinate the stress response.
Yes.
Okay.
And here's where it gets really interesting. They found that this activation of STI1 and the inactivation of ORB6, which promotes cell survival under stress, was also associated with increased lifespan in their model organism. This suggests that manipulating these pathways could hold promise for extending lifespan in other organisms, maybe even humans.
So by understanding how cells respond to stress, we might be able to tweak those responses to make them more resilient and potentially extend lifespan.
That's the idea. It's like fine-tuning the stress response system to make it more efficient and effective.
That makes sense.
And it opens up this whole new avenue for developing interventions that could target these pathways to improve cellular resilience and longevity.
This is incredible how much complexity there is at the cellular level.
Absolutely.
Speaking of complexity, this next paper adds yet another layer to the story of brain health and aging.
Okay.
This one was published on March 4th in Physiology by researchers at Brown University, and the title is Mutation of an Insulin-Sensitive Musical. Drosophila insulin-like receptor mutant requires methionine metabolism reprogramming to extend lifespan.
Okay, so this one might seem a little bit dense at first glance, but it touches on a couple of really important concepts in aging research. The insulin signaling pathway and methionine metabolism.
Right.
So the insulin signaling pathway, as you know, is a key player in regulating metabolism and growth.
Right.
And it's been shown to play a role in aging.
Right.
And then methionine metabolism.
Hmm.
That's the process by which our bodies use the amino acid methionine to build proteins and other essential molecules.
Okay, so how do these two concepts relate to each other, and what did this study find?
Okay, so they used fruit flies, which are a great model organism for studying aging,
Okay.
because they have relatively short lifespans and they share a lot of the same aging pathways as humans.
Got it.
And what they did was they created a specific mutation
Oh.
in the insulin receptor gene in these flies.
Okay.
Now, what's interesting about this mutation is that it extends the fly's lifespan,
Okay.
but without causing them to become insulin resistant,
Interesting.
which is usually what happens in these kinds of studies.
Right? When you mess with the insulin signaling pathway, you usually see changes in insulin sensitivity.
Right.
So what's different here?
What they found was that this mutation actually reprograms methionine metabolism in a way that mimics the effects of dietary methionine restriction,
Okay.
which is a known strategy for extending lifespan
Right.
in various organisms.
So it's like this mutation is hijacking a pathway that's already associated with longevity.
Exactly.
Yeah.
So it suggests that there might be ways to manipulate methionine metabolism, maybe through targeted therapies or even dietary interventions,
Right.
to achieve similar lifespan extending effects without the negative consequences of messing with insulin sensitivity.
That's fascinating how these different pathways in the body
It is.
can interact and influence aging. It highlights the need to look beyond single targets and consider this whole network of interactions.
Absolutely. Aging is complex, and understanding the interplay between these different pathways
Right.
is key for developing effective interventions.
Okay, let's move on to a paper that really challenges our assumptions about a protein typically associated with telomeres.
Okay.
You got it. The paper TRF2, non-telomeric function, is indispensable for neural stemness, from the Council of Scientific and Industrial Research Institute of Genomics and Integrative Biology.
Okay.
CSR-IGEB
Okay.
really makes us rethink our understanding of TRF2.
Mm-hmm.
It was published on March 4th in Molecular Biology. So as you know, telomeres have long been linked to aging,
Right.
and TRF2 is this protein that plays a role in protecting telomeres.
Yes.
But this study is really revealing that TRF2 has another surprising role,
Okay.
keeping neural stem cells healthy
Wow.
and promoting the growth of new brain cells.
Okay.
Remember those hypothalamic neural stem cells we were talking about?
Yes.
Well, this research suggests that TRF2 might be key for their ability to function properly.
Interesting.
So this is huge because it's suggesting that we may need to rethink how we understand how TRF2 works
Right.
and its implications for aging. It might not just be about telomere length,
Yep.
but also about its role in maintaining the health of stem cells,
Yeah.
particularly in the brain.
Yeah, that's a big change.
It is.
Yeah.
It's a reminder that there's always more to learn,
Yeah.
and that even familiar players in the aging game can have hidden talents.
Right.
Speaking of hidden talents, what if something as simple as sound waves could hold the key to reversing aging?
Interesting.
A paper entitled Low-Frequency Ultrasound Reverses Multiple Characteristics of Cellular Senescence suggests just that. Now, this is an area of research I find really exciting.
Yeah.
This study was published on March 3rd in Advanced Healthcare Materials,
Okay.
and it explored the effects of low-frequency ultrasound on senescent cells.
Interesting.
And the findings were pretty amazing.
Yeah.
They found that this non-invasive technique could actually rejuvenate senescent cells,
Okay.
reversing a bunch of age-related characteristics.
Wow.
They saw improvements in 15 characteristics,
Wow.
from reducing inflammation to increasing telomere length.
That's a lot.
It's a real testament to the power of sound
Yeah.
and its potential for therapeutic applications in the field of aging.
That's pretty cool.
It's like giving those aging cells a sonic spot treatment.
Right.
Now let's shift gears and dive into some research that highlights the power of blood-based biomarkers.
Okay.
Two papers this week really stood out in this area. The first one, clinical progression of clonal hematopoiesis is determined by a combination of mutation timing fitness and clonal structure.
Yeah.
Comes from the Mayo Clinic and was published on March 2nd in Genetics.
Okay.
So this research is really delving into this world of clonal hematopoiesis, which is this condition where certain blood cell populations that are carrying specific mutations expand as we age.
Right.
And this has been linked to a bunch of age-related diseases
Yeah.
and even mortality.
Okay.
So these researchers developed this new metric, which they're calling MACS 120.
Okay.
And it combines information about the timing of the mutation acquisition, clonal fitness, and other factors. And they found that this metric actually outperforms traditional methods for predicting
Okay.
the future growth of these clonal populations
Right.
and their associated health outcomes.
Interesting.
It's like having a crystal ball
Yeah.
that can tell you how these mutations
Right.
might impact your health as you age.
That's cool.
The second paper, Dynamics of Blood NAD and Glutathione in Health Disease Aging and Under NAD Booster Treatment, comes from the University of Helsinki and was published March 2nd in Pharmacology and Toxicology.
Okay.
And this paper is exploring how analyzing blood levels of these two key molecules, and NAD and glutathione,
Okay.
can reveal what they're calling redox fingerprints
Okay.
that are unique to different diseases.
Interesting.
So these fingerprints provide really valuable insight into a person's metabolic health and even their response to treatment.
Oh, that's neat.
It's another great example of how analyzing blood can give us this window into these complex processes of aging and disease.
Right.
It's like having a personalized metabolic roadmap.
I like that.
Okay, so before we move on to the next segment of this deep dive,
Mm-hmm.
are there any papers from this batch that really stood out to you?
Oh, yeah, definitely.
Okay.
I think the paper about the silent X chromosome was so interesting. Aging activates escape of the silent X chromosome in the female mouse hippocampus. It was published on March 5th in Aging.
Okay.
And it's just so fascinating because it delves into one of the long-standing mysteries of aging.
Okay.
Why do women tend to live longer than men?
It's a question that has puzzled researchers for decades.
Mm-hmm.
As you know, females have two X chromosomes while males have one X and one Y.
Right. And one of the X chromosomes in females is typically inactivated early in development.
Right.
But the study shows that aging can actually disrupt this inactivation,
Right.
leading to the expression of genes on the previously silent X, at least in the hippocampus of these mice.
Interesting.
So this is a really novel finding, and it raises a lot of questions about how this reactivation might impact brain function and aging.
It's like suddenly having access to this whole new library of genes. So what are the implications of this for you as a researcher?
Well, I think it opens up a whole new avenue for investigating sex differences in aging and for developing targeted interventions for age-related cognitive decline.
Right.
Imagine if we could harness this reactivation process to protect the brain from the damaging effects of aging.
Right.
It's a really exciting possibility.
It really could change our understanding of how sex chromosomes influence aging.
Mm-hmm.
It's a good reminder that we need to consider these sex-specific differences when we're developing therapies and interventions.
Absolutely.
Now, speaking of interventions, let's shift gears and talk about lifestyle factors and their impact on longevity.
Okay.
A paper was published on March 4th in Nutrition, Metabolism, and Cardiovascular Diseases titled Associations of Composite Dietary Antioxidant Index with Premature Death and All-Cause Mortality, a cohort study.
Okay.
And this one caught my eye because it reinforces the importance of diet in healthy aging.
Yeah, this is a great example of how lifestyle choices can really have a tangible impact.
On lifespan and health.
Right.
The researchers analyzed data from a large cohort study,
Okay.
and they found that people who consume more antioxidants, those molecules that help protect our cells from damage, have a lower risk of dying prematurely.
Okay.
It's a good reminder that what we put into our bodies is just as important as any fancy therapy.
As a longevity researcher, I'm sure you appreciate the importance of a holistic approach.
Absolutely. It's not just about developing new drugs and therapies.
Right.
It's about understanding how lifestyle factors can contribute to healthy aging.
Absolutely. And speaking of lifestyle factor,
Yeah.
let's not forget about the power of exercise.
Right.
A paper entitled Resistance Exercise Training in Older Men Reduces ATF4-activated and senescence-associated mRNAs in skeletal muscle provides some compelling evidence for the benefits of strength training.
Okay.
This study was published on February 27th in Geroscience, and it focused on older men and found that resistance exercise training
can actually reduce markers of aging in muscles.
Okay.
That's promising.
It is.
It supports the idea that staying active can help slow down the aging process,
Right.
at least in muscles. But let's not forget about another crucial aspect of aging research,
Right.
understanding the aging process in different tissues and organs.
Yeah.
A paper published on February 27th in BioArchive, a genome-wide CRISPR screen reveals novel determinants of long-lived plasma cell secretory capacity. From the University of Arizona caught my eye because it delves into this world of plasma cells, those antibody-producing factories of the immune system.
Right.
So plasma cells are essential for immunity,
Yes.
but like many other cell types,
Yes.
they decline with age, leading to a weakened immune response.
Right.
So this research used this clever screening technique to identify factors that influence the lifespan and function of plasma cells.
Interesting.
And what's really exciting about this approach is that it could lead to the identification of new targets for therapies that could boost immune function in older adults.
That would be huge.
It would. So it's like finding ways to keep those antibody factories humming along.
Yeah.
Now, a paper published February 28th in BioArchive from City University, disturbed proteostasis accelerates cellular senescence through lamin B receptor dysfunction.
Sure.
Also caught my eye because it challenges this traditional view of cellular senescence.
Right.
That process where cells stop dividing and start pumping out these inflammatory signals. We often think of senescence as this passive process.
Right, like cells are just given up.
Yeah, like they're retiring.
Yeah.
But this research suggests that it's actually much more active
Oh, okay.
and regulated
Okay.
than we previously thought.
Interesting.
They found that disruptions in proteostasis,
Okay.
that intricate system that maintains protein balance in cells,
Right.
can actually accelerate senescence.
So it's like throwing a wrench in the machinery.
It is. It's causing those cells to age prematurely.
Wow.
So if we can understand how proteostasis is disrupted during aging,
Right.
we might be able to develop interventions
Yeah.
to maintain that balance and delay senescence.
That makes sense.
And that could have huge implications for treating a wide range of age-related diseases.
Absolutely.
It's a reminder that we need to look beyond those traditional hallmarks of aging
Right.
and consider these other processes
Yeah.
like proteostasis
Yeah.
that might be playing a role.
It's all connected.
It is. This study is opening up new avenues for research and for developing therapies
Right.
that target proteostasis to combat aging.
Interesting.
It's like finding the fountain of the youth in our own cells.
I like it.
Now, another paper that stood out to me was the common hallmarks and interconnected pathways of aging, circadian rhythms, and cancer. Implications for therapeutic strategies.
Right.
This was published on February 27th in Research,
Okay.
and it explores these intricate connections
Yeah.
between three major players: aging, circadian rhythms, and cancer.
Interesting.
So this one is really interesting because it highlights the interconnectedness
Yeah.
of these seemingly disparate processes.
Right.
So the paper delves into the shared hallmarks of these three processes,
Okay.
such as genomic instability, cellular senescence, and chronic inflammation.
Right.
It also explores how disruptions in circadian rhythms,
Yes.
that internal clock that regulates so many bodily functions,
Right.
can increase the risk of both aging and cancer.
That makes sense.
It makes you think about how important it is to maintain a healthy circadian rhythm.
Yeah.
You know, getting enough sleep, eating at regular times, being exposed to natural light.
Absolutely.
These are all things that can help keep that internal clock in sync.
Mm-hmm.
But what are the implications of this research for someone working in longevity biotech?
Well, it suggests that targeting circadian rhythms could be a new strategy for both cancer treatment and anti-aging interventions. For example, we might be able to develop drugs
Right.
that modulate circadian clock genes to improve sleep, reduce inflammation,
Right.
and even slow down the aging process.
It's like hitting two birds with one stone.
Yeah.
Now let's talk about a paper that explores a potential link between aging and fertility in females.
Okay.
Multiomics reveal the metabolic changes in cumulus cells during aging, published on March 4th in Cell Proliferation.
Okay.
This is another area of aging research that often gets overlooked.
Right.
So this study takes a deep dive into the molecular changes that occur in cumulus cells,
Okay.
those cells that surround and nourish developing eggs
Right.
as females age.
Okay.
So they used a combination of techniques,
Mm.
including proteomics and metabolomics,
Okay.
to create this really comprehensive picture of these changes.
Interesting.
And what they found was that certain metabolic pathways,
Okay.
particularly those involved in energy production
Mm-hmm.
and amino acid metabolism,
Okay.
are significantly altered in aged cumulus cells.
Interesting.
And we know that these changes can have a negative impact
Yeah.
on egg quality and fertility
Okay.
as women age.
Right.
So could these findings lead to new ways to preserve fertility in older women?
I think that's the hope.
Okay.
And what's particularly interesting is that they found that supplementing with serotonin, which is a neurotransmitter
Right.
that plays a role in mood and sleep, but
Yeah.
also has these other functions in the body, could actually improve the metabolic function and meiotic competence of eggs from older mice.
Wow. So could serotonin be a potential key to preserving fertility
It's possible.
as women age.
Yeah, it's a really intriguing finding, and it opens up a whole new line of research
Right.
in reproductive aging.
Absolutely. It's a reminder that the aging process affects every part of our bodies,
Yeah,
even those involved in reproduction.
right.
Now, before we wrap up,
Okay.
I want to touch on two more papers
Okay.
that delve into these molecular mechanisms of aging.
Sure.
The first one, hypoxia-inducible factor and cellular senescence in pulmonary aging and disease, was published on March 4th in Cellular Senescence.
Okay.
And it focuses on the lungs
Mm-hmm.
and how these two key processes, hypoxia-inducible factor signaling, or HIF signaling,
Okay.
and cellular senescence,
Mm.
contribute to aging and age-related lung diseases.
Okay.
So remind me again what HIF does and why it's relevant to aging.
Okay, so HIF is this master regulator that helps cells adapt to low oxygen levels,
Right.
a condition known as hypoxia.
Okay.
And it plays a role in a bunch of processes,
Right.
including angiogenesis, which is the formation of new blood vessels,
Right.
and energy metabolism.
Okay.
Now, as we age, our tissues become less efficient at delivering oxygen.
Right.
So HIF signaling becomes increasingly important.
So HIF helps cells cope with the stress of low oxygen.
Yes.
We know that cellular senescence, those retired cells,
Yeah.
also plays a role in aging.
Right.
So how do those two processes interact in the context of lung aging?
Right. Well, that's what this study was trying to figure out.
Okay.
They found that HIF signaling and cellular senescence are really closely connected,
Okay.
and that their dysregulation can speed up lung aging and the progression of diseases like COPD and IPF.
It's a reminder that aging is this complex process
Yes.
that involves this delicate balance of various pathways.
Right.
And disruptions in this balance can have big consequences
Yeah.
for our health.
Absolutely.
What was the second paper you wanted to discuss?
The next one was incongruent end structures of leading and lagging telomeres dictate the nature of end replication problem.
Okay.
It was published on February 27th in bioRxiv.
Okay.
And this one takes a deep dive into the world of telomeres and the end replication problem.
Telomeres. Those fascinating structures that protect the ends of our chromosomes.
Yes.
And as you know, the end replication problem refers to the fact that DNA polymerase,
Right.
the enzyme that copies DNA,
Mm-hmm.
can't fully replicate the very ends of linear chromosomes.
Right.
So this leads to a gradual shortening of telomeres with each cell division.
Right. A hallmark of aging.
Right.
Yeah.
So this study offers some new insights into the molecular details of this problem.
Oh.
They used a clever model system in yeast to study telomere replication
Okay.
and discovered that the leading and lagging strands of the telomere actually have distinct end structures.
Oh, wow.
So the two strands of the DNA molecule at the telomere are handled differently during replication.
Yeah, that's interesting.
So they found that the lagging strand telomere has a short overhang,
Okay.
while the leading strand telomere has a blunt end
Okay.
that's protected by this protein complex.
Okay.
So this asymmetry
Right.
has implications for how the telomeres are replicated and maintained.
Wow.
And what's particularly interesting is that they found this specific enzyme, RNase 2,
Okay.
plays a crucial role in removing the last RNA primer
Okay.
from the lagging strand telomere during replication.
Interesting.
Now, when this enzyme is absent, the RNA primer is retained.
Expert, it actually helps to protect the telomeres from erosion and delays senescence in cells that lack telomerase.
This is huge because it's suggesting that manipulating RNase H2 activity could be a potential strategy
Yeah.
for influencing telomere length and cellular lifespan.
That's really cool.
It's a reminder that even the most fundamental processes of DNA replication can have profound implications for aging.
It's all connected.
It is.
Oh,
Okay, we've covered a lot of ground in this deep dive.
we have.
Let's take a moment to reflect on what we've learned and discuss the broader implications of these findings.
Okay.
So it's amazing to see how much progress is being made in the field of longevity research, from reversing senescence
Yeah.
to rejuvenating the hypothalamus to manipulating circadian rhythms and even potentially preserving fertility.
Right.
These papers offer a glimpse into a future where aging might not be the inevitable decline we once thought it was.
I completely agree. It's a really exciting time to be working in this field. There's so much momentum and this feeling that we're on the cusp of some major breakthroughs that could really fundamentally change how we approach aging and age-related diseases.
And what's so striking to me is the diversity of approaches being explored.
Right.
You know, we've seen everything from cutting-edge genetic techniques to lifestyle interventions to the surprising potential of sound waves.
Yeah, it's amazing.
It seems like researchers are leaving no stone unturned in their quest to understand and manipulate the aging process.
Absolutely. And I think that speaks to the fact that this is a truly multidisciplinary field.
Right.
We're drawing on expertise from biology, genetics, medicine, computer science, engineering, even physics.
It takes a village.
It really does.
Yeah.
And I think that this diversity of perspective is really what makes it so dynamic and innovative.
So as a biotech researcher listening to this,
Yeah.
what do you think are the most important takeaways from these papers? What should our listeners be paying attention to?
I think the biggest takeaway is that the field is ripe with opportunity.
Okay.
We're moving beyond simply treating the symptoms of aging,
Right.
and we're starting to address the root causes.
Right.
And I think this shift in focus is opening up these incredible possibilities for developing therapies
Right.
that can actually target the very processes that drive aging.
Right.
The papers we talked about today highlight some very promising avenues, from manipulating specific genes and pathways
Mm-hmm.
to harnessing the power of stem cells to understanding this complex interplay
Yeah.
between aging, circadian rhythms, and even cancer.
And it's not just about extending lifespan.
Right.
It's about extending health span,
Yeah.
those years of healthy, vibrant life.
Yeah.
These findings are pushing us closer to a future where aging doesn't have to mean decline,
I agree.
where we can all live longer, healthier, more fulfilling lives.
Absolutely. And it's important to remember that this research isn't just happening in a vacuum.
Right.
It has the potential to translate into real-world solutions, into therapies and interventions
Absolutely.
that could improve the lives of millions of people.
So to our listeners out there on the front lines of longevity research,
Yeah.
keep pushing the boundaries, keep exploring, keep asking those tough questions. The answers you find could truly change the world.
Couldn't agree more. And one final thought to ponder. If aging is truly a malleable process,
Okay.
what will you do to optimize your own longevity?
Okay.
What lifestyle choices will you prioritize? What areas of research will you focus on?
Right.
The future of aging is in our hands,
Yeah.
and it's up to us to shape it.
That's a powerful message to end on. Thanks for joining us for this deep dive into the longevity papers. We'll be back next week with another batch of groundbreaking research. Until then, stay curious and keep exploring. Wow, it is so amazing to see all this progress being made in longevity research, from reversing senescence to rejuvenating the hypothalamus to manipulating circadian rhythms and even potentially preserving fertility.
It's really exciting.
These papers really, offer a glimpse into a future where aging might not be the inevitable decline that we once thought it was.
I completely agree. It's an incredibly exciting time to be working in this field. And there's just this real sense of momentum, this feeling that we're on the cusp of some really, major breakthroughs that could fundamentally change how we think about aging and age-related diseases.
And it's not just, about extending lifespan.
Right.
It's about extending health span.
Yeah.
Those years of healthy, vibrant life.
Yeah.
And these findings are really, pushing us closer to a future where aging doesn't have to mean decline.
Yeah.
And where we can all, live longer, healthier, and more fulfilling lives.
I agree. And I think it's important to remember that all this research isn't just happening, in a vacuum.
Right.
It really has the potential to translate into real-world solutions,
Yeah.
into therapies and interventions that could improve the lives of, millions of people.
Absolutely.
Yeah.
So to all of our listeners out there, on front lines of longevity research,
Yeah.
keep pushing the boundary, keep exploring, keep asking those tough questions. The answers that you find could truly change the world.
Absolutely. And, one final thought to ponder. If aging is truly a malleable process,
Okay.
what will you do to optimize your own longevity? What lifestyle choices will you prioritize? What areas of research will you focus on? The future of aging is really in our hands.
Yeah, it is.
And it's up to all of us to shape it.
That's a powerful message to end on. Thanks for joining us on this deep dive into the longevity papers. We'll be back next week with another batch of groundbreaking research. Until then, stay curious and keep exploring.